Hello students, welcome to my channel Advanced Botany. First of all, I would like to thank all for your time and continuous support. Today, I look forward to discussing photoprotective mechanisms in plants. In our previous lecture, I have already told you about 4 photoprotection mechanisms. First is light avoidance associated with the movement of leaves and chloroplast. Second is screening of photoradiation and synthesis of antioxidant molecules and enzymes. Third is quenching of singlet and triplet excited states of chlorophyll. And fourth is repair processes for damaged photosystem 2 and photosystem 1 reaction centers. First two mechanisms we have already discussed in our previous lecture. Today we will discuss third and fourth mechanisms, that is quenching of singlet and triplet excited states of chlorophyll and repair processes for damaged photosystem 2 and 1 reaction centers. First, I will tell you about chlorophyll and chlorophyll triplets. Chlorophyll is the main light absorbing pigment in the light harvesting complex, in the inner antenna, and also in the reaction center. Chlorophyll is very efficient in absorbing light and has the additional advantage that the excited states are long-lived enough to allow the conversion of the excitation energy into the electrochemical potential via charge separation. If the energy is not efficiently used, the spins of the electrons in the excited state can rephase and give rise to a lower energy excited state known as chlorophyll triplet state. This chlorophyll triplet state uh, has an even longer lifetime and can react with triplet oxygen to produce a very reactive singlet oxygen if no efficient quenchers are around. So triplet chlorophyll state formed either directly or by charge recombination. Just look at this diagram. When light falls on chlorophyll molecules in antenna or in reaction center, it gets excited and moves to higher energy state called excited singlet state. Then this chlorophyll molecule transfers energy to another chlorophyll molecule and itself returns back to its ground state. Now, triplet chlorophyll is formed when excess light energy harvested by chlorophyll cannot be dissipated efficiently. So, triplet chlorophyll and triplet P680 molecules are formed. This triplet chlorophyll then react with triplet oxygen and it gives rise singlet oxygen species, right? So this singlet oxygen is a problem because singlet oxygen, which is formed from chlorophyll triplets, can react with proteins, pigments, and lipids, and is thought to be the most important species responsible for light-induced loss of photosystem 2 activity. Also, the degradation of D1 protein. D1 protein in the reaction center of photosystem 2 and for pigment bleaching. So, singlet oxygen formation is favored under certain physiological conditions like exposure to high light intensities or drought leading to closure of the stomata and low carbon dioxide concentration in the chloroplast. So under such condition, the plastoquinone pool can be in a very reduced state and forward electron transport is very limited, and then recombination reaction in photosystem 2 can occur. So triplet chlorophyll uh, is formed either directly by intersystem crossing from the singlet excited chlorophyll or by charge recombination reactions in the reaction center. Now, before discussing quenching of singlet and triplet excited states of chlorophyll, I will tell you how this triplet chlorophyll is formed by charge recombination in the reaction center of photosystem 2. In the reaction center of photosystem 2, the first detectable radical pair formed after excitation of light is P680 plus pheophytin, with P680 being the primary electron donor and pheophytin the primary electron. The next step of electron transfer after the formation of the primary radical pair leads to the reduction of primary quinone acceptor, QA. Subsequently, P680 is reduced by electron donation from the redox active tyrosine, which itself obtains an electron from the water oxidizing complex. So charge recombination reaction occur when the forward electron transport cannot proceed. Let's just look at this diagram. Here you can see this is a linear electron flow, means this is a normal electron flow from photosystem 2 to cytochrome B6F complex and then to photosystem 1. If this forward electron transport is blocked, then charge recombination reactions occur in photosystem 2. This leading to the formation of triplet chlorophyll, which reacts with oxygen to form singlet oxygen, right? Means if primary quinone acceptors stays reduced because of the block of forward electron transport, the yield of the primary charge separation is lowered. Now, in the closed reaction center, however, if primary charge separation occurs, it is followed by recombination of the charges. So charge recombination in the primary pair will produce either the singlet state, ground state of P680 or triplet state of P680. Under reducing conditions, QA becomes doubly reduced, thereby releasing the negative electrostatic effect on the energy of the primary pair and a high yield of charge separation. Recombination and P680 triplet formation is observed. The double reduction of QA and the high yield of P680 triplet P680 formation in such centers have been suggested to have some means relevance to photo inhibition. The occurrence of double reduced QA, however, has never been shown to occur under means normal physiological conditions. At cryogenic temperature around means it's different from the situation at ambient temperature. The primary radical pair is formed with a high yield irrespective of the redox state of QA, and the yield of the triplet state means the triplet P680 is high both with QA and QEH2. So triplet decay is much faster with QA than Q2H2. Significant amounts of singlet oxygen is produced in photosystem 2 with large antenna under continuous illumination, and they are most likely linked to chlorophyll triplet formation by charge recombination of the primary pair in photosystem 2 and not to chlorophyll triplet formation in the antenna. So, charge recombination of the primary radical pair leads to the production of singlet and triplet P680. Then chlorophyll triplet state can react with triplet oxygen and leading to the formation of singlet oxygen, which will damage the reaction center. So singlet oxygen is a problem. Now, quenching of chlorophyll triplet state and photoinhibition of photosystem 2. Singlet oxygen can react with proteins, pigments, and lipids and lead to the loss of photosystem 2 activity, the degradation of D1 protein, and for pigment bleaching. So, dangerous triplet state of chlorophyll, which is responsible for the formation of singlet oxygen, which is means can be quenched directly by carotenoids in close proximity. Now, in the reaction center, the distance between the carotenes and the triplet chlorophyll is too large to allow the direct triplet quenching. Hence, the primary function of the beta carotenes is probably the quenching of singlet oxygen produced by a triplet state of P680. Now, another important antioxidant located in the thylakoid membrane is alpha tocopherol. Tocopherol is an efficient sequencer which becomes oxidized when reacting with singlet oxygen. If singlet oxygen produced via chlorophyll triplet formation in the reaction center is not quenched by carotenoids or tocopherol, then it is very probable that it reacts with D1 protein as a target molecule. So rapid turnover of D1 protein occurs even at low light intensities, indicating that there is always some singlet oxygen formation even under low or moderate light. Singlet oxygen generating chemicals produce the specific fragments of D1 proteins. The degradation of D1 protein may be regarded as a physiological defense system to prevent uncontrolled damage of photosystem 2. So controlled destruction of D1 protein seems to be an attractive safety valve to detoxify singlet oxygen directly at the place of its generation. Second mechanism is repair processes for damaged photosystem 2 and photosystem 1 reaction centers. Photo-damaged photosystem 2 undergoes repair in a multi-step process involving first is reversible phosphorylation of photosystem 2 core subunit. Second is monomerization of lateral migration of photosystem 2 core from grana to stroma thylakoids. Then partial disassembly of photosystem 2. Next is proteolytic degradation of damaged D1 proteins. Then replacement of damaged D1 proteins with new copy and reassembly of photosystem 2 monomers and migration back to grana thylakoid for dimerization and supercomplex assembly. So these processes require the light-dependent specific phosphorylation of photosystem 2 core proteins. Photosystem 2 core proteins D1, D2, CP43 and PSBH. PSBH, photosystem center protein, photosystem 2 center protein H. Now, two types of chloroplast proteases act cooperatively in the degradation of D1 protein from CP43 free photosystem 2 monomer lacking the OEC. After degradation of damaged D1 protein, a new copy of D1 is inserted into CP43 free photosystem 2 monomers by de novo synthesis. After replacement of the photo-damaged D1 protein by a newly synthesized copy, CP43 is reassembled to form photosystem 2 core monomer, still lacking the OEC. After reassembly of OEC, repaired PS2 monomers need to migrate back into stacked membrane regions for further assembly into dimer and supercomplexes. Thanks for watching. If you like this video, please make sure to subscribe for more. Thank you.